We're going to start with Natalia Ivleva from the Technical University of Munich. And as you can see, she was our Bunsen-Kirchhoff Award in 2020. And as you all know, due to the pandemic situation, we were not able to have her talk in presence. So that is why we would like to give her today the opportunity to present her work and to show new results as well. And it's a great pleasure for me to have you here. Natasha, please, the floor is yours.
Thank you very much, Kerstin, for the kind introduction. And I have to say it was my great pleasure and great honor to receive Bunsen-Kirchhoff Award 2020. And I'd like to thank a German working group for analytical chemistry, Deutsche Arbeitskreis für analytische Chemie, GDCH, for this award, and Analytica AG for the endowment. So, dear all, I'm really happy to see you now in presence and not in online session. So, in my research, I focus on the application of Raman-based method for the analysis of different environmental and industrial samples. But today, I would like to show you some examples for applicability of Raman microspectroscopy for the environmental analysis. So I will start with short introduction in Raman microspectroscopy, and then we'll show how this method can be applied for the analysis of microplastic and nanoplastic, and for the study of microorganisms and biofilms using stable-isotope Raman microspectroscopy, surface-enhanced Raman scattering, and resonance Raman spectroscopy. And then summary and outlook will come. So, as you definitely know, Raman microspectroscopy is a combination of Raman spectroscopy and optical microscopy, which allowed us to get spatial resolution down to 1 micrometer or even below, so down to 300 nanometers. This method is based on inelastic light scattering and provides us vibrational fingerprint spectra. We can perform analysis non-destructively and without interference of water. We can do 2D, 3D images. So, of course, nobody is perfect. Also, this method has some disadvantages, its influence of fluorescence, but in many years we learned how to solve this problem. So, let us start with the analysis of microplastic and nanoplastic. These particles became emerging particulate anthropogenic pollutants. And from analytical point of view, it's really complex analyte. So we have broad size range for microplastic from 1 micrometer to 1,000 micrometer, and particles smaller than 1 micrometer are defined as nanoplastic. We have different polymer types, so polyethylene, polypropylene, polystyrene, and so on. We have also different shape of these particles, from spherical particles to fibers and some films or foams, which makes automated recognition of these particles really challenging. We have also various additives and also sorbed contaminants on the surface. Plastic particles can be also in different aging state. It can be primary particles, so intentionally produced, or secondary particles, which are formed in the environment due to fragmentation and degradation. And this complexity may explain why the degree of contamination of the environmental and food, especially with small particles, still unclear. The reported values for microplastic range in 10 order of magnitude, so from less than 1 particles and up to 100 million particles per cubic meter. And for nanoplastic, even less reliable data exist. Therefore, it's real need to develop reliable method for the analysis of small microplastic and nanoplastic particles. Only then we will be able to find the needle in the haystack. So generally, methods which are applied for the analysis of microplastic and nanoplastic can be divided in 2 groups. It's mass-based method, which provides us information about mass of microplastic and nanoplastic particles. These methods are really important if we want to get data for modeling or for some regulatory reason. But if we have particles from toxicological point of view, it's really important to know the number of particle size distribution, shape of these particles. And for this, particle-based method are applied. And here we, of course, focused on Raman microspectroscopy. So from analytical point of view, we know that analysis of 1 or 2 particles will not be sufficient. And normally we have a lot of other particles on our filter. Sometimes we can have even up to 1 million particles on one filter. And here's the question, how many particles have to be analyzed to make this analysis analytically sound? And to answer this question, we applied random sampling without replacement method. It's some statistical method which is used also for the prediction of election results. And here's the number of particles which we have to measure depend on the entire number of particles, on the estimated microplastic fraction, and of course margin of error, so error which we can accept for the analysis. And as we can see, even if we have up to 1 million particles with 1% of microplastic fraction, it will be sufficient to measure 5 or in this case 7,000 particles to reach analytically sound results. So if we know how many particles we have to measure, how we choose these particles on the filter. So here different procedure can be used with cake model or snail model, where windows are chosen at some systematic procedure, but we know that representative subsampling can be really realized if we do random sampling, and we decided for this method in our analysis. So for a couple of years, we started with the vision, so it was Elisabeth von der Esch who applied this method. We start with the vision, how we want to analyze the particles. So we wanted to deposit all particles and fibers on the filter, then localize these particles to have information how many particles in total we have on the filter, then choose the number of particles which have to be analyzed and number of fibers, then perform Raman analysis, and in this case, combine morphological and chemical information. And this procedure was already realized and published 2 years ago in form of open source software, TUM-ParticleTyper. This method allowed for detection, characterization, and quantification of particles and fibers on the images. It can be depth field Raman microscopy images, but it's also suitable for the analysis of scanning electron microscopy images and fluorescence images, because we have red, blue, and green channels. And if we have then information about particles on our filter, we can identify randomly chosen fragments with Raman microspectroscopy. And with this method, we were able to analyze up to 7,000 randomly chosen fragments, bigger than 10 micrometers on the entire filter, so for representative analysis. And since all procedure now run automatically, we also call this program TUM-Party, because student can then do something different. So, till now, everything good. But what about particles smaller than 10 micrometer? We know that with Raman microspectroscopy, we can go down to 1 micrometer or even below. Can we do it automatically? In principle, yes. But if you want to measure entire image from the filter, you need high magnification objective, and your image will be huge from the data point. And also, if you first have huge image and then try to match 1 or 2 micrometer particles, it can be a problem due to not really precise motorized table, or if filter change a little bit. Therefore, we develop here first theoretically, so Christian Schwaffert was here in charge. He developed theoretically first procedure for randomly chosen windows, and then we choose the window, characterize the number of particles, size of particles, and then perform Raman analysis, and we go to the next window, and so on. But in this case, we do not know information how many particles we have on the entire filter. So it means that we cannot apply classical procedure to calculate our error. And in this case, we can also use statistical methods, so bootstrap, where, for example, if we measure 50 windows, we can accept this subsampling as sample and do Werthal mixing. And if we have relative similar distribution of particles on the whole of these windows, our standard deviation will be low. Then we can say our confidential interval is good. If difference is really high, we have to measure additional windows, so we can continue this procedure and everything automatically. So it will be part of our TUM-ParticleTyper 2, which is now in preparation for publication. And it should help us to analyze particles smaller than 10 micrometers. And in this new procedure or new software, we also plan to improve the agglomeration procedure, but it's always the question by particle like this, it's one particles or it's agglomerate. You can choose it as one particle, but you will lose a lot of information if you measure just one point. If you do the agglomeration, for example, with watershed, you can cut your fibers or cut your particles in many particles and will overestimate the number of particles and fibers on your filter. And in this case, we try to optimize it and developed the agglomeration using adaptive watershed. So we compared what curvature particles have to use this deagglomeration procedure, and this deagglomeration is now done automatically to avoid human bias. And so if we combine these two steps for the analysis of entire filter for particles bigger than 10 micrometers and analysis of fragments with window method for size range from 1 to 50 micrometers, so we can have an overlap for our in-house validation. So these methods we applied for the analysis of particles bigger than 1 micrometer. But theoretically, we can also go a little bit down. So according to Rayleigh criterion, particles so down to 300 nanometers can be analyzed, and Christian Schwaffert was able to get separated Raman signal from 2 particles in this size. But to be sure that single particles scanning electron microscopy images were applied. So it's possible to do, but we have to say it's really time consuming and particles have to be separated. Therefore, if you want to perform detailed analysis, it can be applied, but for high throughput analysis, something different has to be found. And we decided to use the combination of field flow fractionation and Raman microspectroscopy for the identification of particles. So with field flow fractionation, we obtained size fraction of particles smaller than 1 micrometer. They can be detected with UV detector, and size information is achieved by multi-angle light scattering detector, and then bulk measurements are performed with Raman microspectroscopy. This field flow fractionation, I hope you know how it works. Here, 2 flows or 2 fields are applied. On the one hand side, it's laminar flow with our particles, and then perpendicular flow is applied. For asymmetrical flow, field flow fractionation, it's flow, and particles can be separated according to their size. First, small particle will come, and then fractions with bigger particles will follow. It's in size range of 1 nanometer to 1,000 nanometer, it can be performed, so we can cover entire nanometer range. But for some application, we found out that centrifugal FFF can be even more suitable because it's allowed to separate the particles by applying of centrifugal field, not only according to the size, but also according to the density, and cover the region from 10 nanometers to 50 micrometers. Here you can see this hyphenation, so online half-plane in reality in our new lab in Garching. So here's asymmetrical low field flow fractionation system, and Raman microscope. So we combine these two systems, but of course, to combine it, we need something what was realized in form of flow cell, and to increase the sensitivity of our Raman signal, because we know that Raman signal is weak signal, we use optical tweezing, 2D optical tweezing with the same laser, usually a 532 laser. We capture the particles and perform Raman analysis. So here's one example how it works if we combined centrifugal field flow fractionation and Raman microspectroscopy. So here's a mixture of polystyrene particles of 350 nanometers and 500 nanometers mixed with PMMA particles of 500 nanometers. Normally, these two fractions of 500 nanometers will come together at asymmetrical field flow fractionation, but with centrifugal FFF, due to different density of these particles, we first observe smaller fraction of polystyrene, then bigger particles. They are even a little bit bigger compared to PMMA particles, but since they are lighter, they come as separate fraction, and third PMMA fraction appears. And you see that with Raman signal, we can assign two first fraction to polystyrene and third fraction to PMMA. So you would say with standards, it's not so difficult to work. Of course, we want to go in direction of real samples. And the first step here, we try to analyze also polydispersed polyethylene particles. So in this case, we cannot observe really separated size fraction, but we see for smaller particles first polystyrene, then polyethylene in next fraction, and at the end, PMMA. So it looks so that we can combine fractionation, sizing, and identification of particles smaller than 1 micrometer. So to shortly sum up this part before I will go to the next part of my presentation, with Raman microspectroscopy, we can perform automated representative analysis of plastic and non-plastic particles and fibers bigger than 10 micrometer. We are working on the development of method to be able to measure automatically fragments down to 1 micrometer. So we found out that combination of Raman microspectroscopy and scanning electron microscopy is really helpful for the detailed characterization of particles. But if we want to go to the high throughput analysis, here hibernation of field flow fractionation and Raman microspectroscopy will be method of choice. So we are definitely not completely ready with the application of this method for real samples. Therefore, we want to optimize and validate our method for the automated analysis down to 1 micrometer for the real samples. So we are working on increasing of sensitivity of chemical analysis of nanoplastic. For this, pre-concentration and enrichment of nanoplastic particles will be crucial. And the next and really challenging step will be quantification of nanoplastic particles. So now I would like to change the topic and discuss applicability of Raman microspectroscopy for the analysis of microorganisms and biofilms. We know that most of microorganisms are living in form of biofilms, and in aquatic systems, biofilms play important role in water quality related substances. And therefore, it's real need for the analysis of microorganisms and biofilms with good spatial resolution and chemical information. And we believe that here, stable-isotope Raman microspectroscopy can provide us information on the accumulation and degradation of pollutants at the 1 micrometer level. So while it works with stable-isotope Raman microspectroscopy, so we know that vibrational frequency is inversely proportional to the reduced mass. And if heavier isotope instead of normal isotopes are applied, we will see vibrational bands at lower frequency, so lower frequency, longer wavelengths. Therefore, these shifts are called redshift, it works perfectly for carbon. For example, Wei Huang shows that if bacteria are cultivated with 13C glucose instead of 12C glucose, the redshift of protein, nucleic acid, lipid bands can be observed, especially sharp phenylalanine band is suitable for the analysis. So, therefore, in our laboratory, we, so best to say, Patrick Kubrick for a couple of years, analyzed sulfate-reducing N47 bacteria, which are able to degrade naphthalene, and we apply fully labeled naphthalene, but found not just one band for fully labeled phenylalanine, but 4 bands which can be assigned for phenylalanine without 13C with 2, 4, and 6 atoms. So it means that we cannot just say that some bacteria are labeled, some not. We can get detailed information on the chemical composition and stable isotope distribution in the biomass. But all of these gray spectra, single spectra, were measured for more than 10 seconds. And it means that it's a really time-consuming analysis. And to increase the sensitivity of this method, so the solution was obvious, we apply surface-enhanced Raman scattering. CS is a method which allowed to significantly increase Raman signal intensity if molecules are close or attached to the nanometer-sized metallic structure, silver or gold. And here at least 2 effects sought to contribute to the total enhancement. It's electromagnetic enhancement or surface plasmon resonance, and chemical enhancement, charge transfer. For electromagnetic enhancement, we need our analyte in close proximity to the CS surface because effect will decline rapidly with the distance. For the chemical enhancement, we need close, or we need direct contact of nanoparticles and our analyte. And one of really suitable CS substrate here is silver nanoparticles, which we produced according to Leopold and Leder procedure at room temperature with silver nitrate and hydroxylammonium hydrochloride. But if you ever try to do CS, you may know that to get enhancement, it's easy. To get it reproducible, it's really challenging. And it becomes even more challenging if you want to get it reproducible at single cell level. So, and to solve this problem, method for in situ preparation of CS substrate was developed at our institute and published by Zhou et al. And then Patrick modified it and applied for analysis of non-labeled and labeled bacteria. And you were able to obtain reproducible peak, reproducible spectra with most pronounced peak at 730 inverse centimeter. And if bacteria were labeled with 13C, we observed the redshift expected, and with 15N, it was even more shift, and most pronounced redshift was observed if 15N and 13C were applied together. And this labeling procedure allowed us to assign this band to the adenine-related compounds because at that time it was not clear what compounds contribute to this signal. And we found that this signal can be only found for metabolically active bacteria. So if bacteria are dead or metabolically inactive, you will get no or very weak CS signal. So with this procedure, it is possible to do rapid discrimination and characterization of bacteria at single cell level. And if we go further, we can analyze also biofilms. Here is artificial biofilms where labeled and non-labeled bacteria were embedded in agarose gel, and it was possible not only localize the bacteria, but also to differentiate labeled and non-labeled cell. And at the end, some really new results which are still in preparation. We hope to submitted soon from Julian Weng and Kara Müller, where we apply stable-isotope Raman approach for the analysis of carotenoid-containing bacteria. So in this case, we can use resonance effect in Raman spectra and observe the bands of carotenoids, which are shifted. And if we combine 13C and deuterium labeling, we found even so non-described shifts. Some of bands are redshifted as expected, but for one band, we even observed some unexpected blue shift. So we are in a stage where we try to explain everything. And with this procedure, we can really track 13C and D uptake to get information about cell growth and metabolic activity simultaneously at the single cell level. So, at the end, short summary, with stable isotope Raman microscopy, it's possible to perform analysis, but if we combine it with SEERS, we can do it really sensitive. It is also applicable for 3D non-destructive in situ analysis of complex matrices, and with multi-element stable isotope resonance Raman microscopy, we can really unravel a lot of processes and get information about single cell heterogeneity. And here we are working further on the applicability of stable isotope approach for the analysis of degradation of microplastic. It's Kara's work. And we also plan to optimize RAM-based 2D and 3D images and combine it with mass-based techniques for sensitive and spatially resolved data. So then at the end, I really like to thank all members of our institute and other colleagues from in other institutions and companies who helped me and my group to perform these results. So my special thanks go to Professor Nisner, who supported me for many, many years, and with his special art, he's helped me to find my way in analytical and environmental chemistry field. So my thanks go also to Professor Elsner, who allowed me to continue my work at the institute, and special thanks, of course, for former and current member of Raman and SAM group. So without them, I wasn't able to present my results here. They helped me to realize my idea and bring new input and help me to go further. Of course, I would like to thank also funding from DFG, from Federal Ministry of Education and Research, and in the end, thank you very much for your attention.
Thank you very much, Natasha, for this really inspiring lecture on Raman and